So our second presentation will pick up on some of the things that Simon talked about. So Eloise Spanner from University of Sydney is going to deliver the second part of the webinar today. So she's a second-year PhD student at the University of Sydney, and she first started working on factors affecting the success of AI in sheep as part of her honours project. So, and she's sort of really interested in the sort of concept of using advanced semen assessment technology and statistical modelling to predict fertility of size for sheep producers. So thanks, Eloise. So I will hand over to you. So share your screen and we'll go into part two.
Thank you, Sue, for the introduction. I'm just checking everyone can see my screen.
Yeah, we can. Thank you.
Perfect. All right. Well, thank you for the introduction. Over the last 2 years nearly, I've been working on this project called the Prediction of Artificial Insemination Success, and I'll give you today a bit of an overview of what we've been doing and a little bit of insight into the project. So I'll give you an overview of what I was talked about today. We'll start with the backgrounds and the aims and objectives of the study. This will lead us into the factors of fertility, experimental design, and the statistical modelling that will be going on. And then we'll go into a bit of predicted outcomes, the importance of the research, and hopefully we get time for some questions. So, artificial insemination, AI, is an advanced reproductive technology used to increase the number of lambs born to elite flock to accelerate genetic gain across the Australian sheep industry. With over 68 million sheep in Australia, only 1% each year are entered into artificial breeding programs. And we question, why do these numbers remain so low when we compare it to the dairy industry, which is nearly 100%? Well, on average, 70% of ewes that are entered into artificial breeding companies are expected to fall pregnant. Unfortunately, these results do vary significantly between programs and can reach lows of 30 to even 10%. So if we can identify what causes this variation in AI success, whether male, female, and environmental factors, we can increase productivity, genetic gain, and financial revenue for the industry. So initially, to assess the fertility of a ram and ewe, you'll begin with a physical assessment. This will look at the 4 T's, breeding soundness, their ability to copulate, age, and body condition score. Those interested in semen quality can then perform a basic semen assessment, and this looks at motility, concentration, and morphology. However, some issues with these assessments are they are subjective and introduce observer bias. And within the industry, there's no real standard reference, and we think this may cause variation in AI results. So, an advanced semen assessment can be performed in a research facility. We can look at sperm quality traits such as velocity, acrosome integrity, viability, plasma capacitation, DNA integrity, membrane oxidative status, as well as proteomics. So this introduces the project we've been doing, and it's to predict if one or a combination of male, female, and environmental predictors significantly correlate to fertility. We hope to select rams and ewes of high fertility potential to identify markers of fertility and to establish a set of semen standards that can lead to greater fertility. Just an overview of the fertility factors we'll be looking at in this study. So we'll begin with the male factors, and we do perform a basic semen assessment looking at motility, morphology, and concentration. We also can be performing advanced semen assessments, so we'll look at viability, acrosome and DNA integrity, membrane and oxidative status, as well as proteomics. The female factors we'll be exploring are age, intra-abdominal fat, uterine tone, estrus synchronization, as well as AI time. And the environmental factors include climatic conditions, nutrition, or if there's been a drought or flooding event around the time of AI. To give you an indication of how this data collection works, we begin with a sheep handler that doses and sedates the ewe. The ewe is then tipped and placed on the crate and moved to the AI bench. Here, the AI tech and vets work together to perform the insemination, and my staff collects all the female data at the time of AI. So this project is a collaboration between the University of Sydney and the Australian Merino Site Evaluation Association, and 7 commercial sites around Australia. We have 400 merino rams that have been collected over 3 breeding seasons and inseminated into about 35,000 ewes. So at the time of AI, this is when we collect all our female data. And so intra-abdominal fat and uterine tone is scored from 1 to 5 by the inseminator. So you can see the first image is a fat of 5 and a tone of 1. So there's an excessive amount of fat in the abdominal lining, and the uterine horns are very flaccid and quite small. And this is compared to a fat of 1 and a tone of 5. And you can see there's very minimal fat along the abdominal walls, and the uterine horns are quite hard and turgid. Other things we measure at AI is the time of AI post-cedar pull, and this is calculated by minusing cedar removal time by insemination time, and this gives us a raw hour time difference between the two. And we can also measure age, and we do this by looking at the EID colour tag associated with the year of lambing, and this will give us an indication of the time of AI. Environmental conditions at time of AI also measured. So we look at temperature, rainfall, humidity, feed, sunlight hours. Again, if there's any big weather events that have happened during the time of AI. So the key to the success of this project is that the ejaculates that are used for the insemination are sent to the university for our semen assessment. So this is a massive day, and it requires nearly our entire lab team to process and assess each ejaculate across 6 hours. We repeat each assessment 3 times at 0, 3, and 6 hours post-thaw. So our post-thaw initial assessment includes volume and then concentration by the nuclear counter, and this gives us the total sperm number per mil.
Eloise, can you just check your microphone? I seem to have lost your audio just for the second.
Is it back? Can you hear me now? Yep. Okay.
Yeah, we can just start from the motility and kinetic parameters, and that way we'll make sure everyone can hear the important bits.
Yeah, of course.
Thank you.
So yeah, the subjective motility is assessed under the phase microscope, and it gives us the percentage of sperm moving forward. Sorry, just percentage of sperm moving. And the CAST is an objective tool that measures motility and kinetic parameters, and it gives us an indication of the percentage of sperm progressively moving forward. We can then now begin our advanced semen assessment, and we use the flow cytometer, and we've been incorporating 5 different stain combinations to assess different sperm quality traits. So the first one we begin with is variability, and this measures membrane integrity. We then have acrosome integrity, which measures acrosome damage if the acrosome is intact. Then we can move on to lipid peroxidation, which looks at membrane stability if the sperm has gone under early capacitation. Mitochondrial superoxide, which looks at intracellular reactive oxygen species of the mitochondrial. And we can also measure the reactive oxygen species of the nucleus, and this is called oxidative stress. And we can go into more detail and measure the DNA integrity of a sperm cell by looking at the percentage of DNA fragmentation. So we've collected our female, male, and environmental data. Now we collect our fertility data by ultrasound, and this is roughly 50 days post-insemination. And we look at the pregnancy status to determine if it's pregnant by AI, a backup ram, or if it's empty. And I just want to reiterate that we're together seeing what combination of in vitro semen predictors, female and environmental factors strongly and significantly correlate to fertility. So we do this statistically by running a generalized linear mixed model. And the first thing we do is, does one predictor alone significantly correlate to fertility? If it does, we combine all those factors into a multivariate model. And here we keep all the significant factors together. We do this by also running PCAs and correlation matrices to see if there's any relationship between the variables. So what can we expect from this program and what do we see happening? Well, we do know there's a massive difference amongst sires and their fertility. And the only way to determine what causes this variation is we need to complete an in vitro semen assessment. We also know that synchronization plays an important role in fertilization, as it influences the time of ovulation and that influences the time of insemination. As well as changes to climates, we need to make sure that the ewes and rams are in best condition, and this may relate to body condition and the overall health and well-being of a ewe before and after the breeding program. So a data set of this size and magnitude is yet to exist for a sheep artificial breeding program. So by combining in vitro semen assessments with novel technologies to field fertility results in one large data set, we hope to consider all male, female, and environmental factors that can help explain pregnancy success. So that's the project, but how does this relate to the industry? What's its importance? So ultimately, this program aims to reduce infertility to increase the number of pregnancies. Therefore, with more lambs born to elite flocks, we can accelerate genetic gain across the Australian sheep industry. This project involves a collection of people, and I have to acknowledge Dr. Jessica Rickard and Professor Simon de Graaf for their support, and AWI for the funding of this project, as well as all the industry bodies that have given us their support over the many years for this project. Without them, it wouldn't exist. So I continuously thank them for their time and support. And now I will open up questions if we have any questions in regard to the project. Thank you for listening.
That's great, Eloise. Thank you for a really good overview of that project. And it's a really, like you say, it's a really exciting database. It's large, covering different seasons, different geographical areas. So it really should help us to answer those sticky questions related to.
Hopefully.
I guess just to start it off, I'll just make sure I can see everyone's questions. Yeah, and that's sort of a bit of a follow-up question first, sort of from one of our attendees. Is there any scope, because we have this database and because you're working with AMSIO, there should be some scope to look at how the size, genetics, and how its performance or other traits, how they're linking together with some of these sort of sperm traits?
Yeah, so are you talking in relation to like the sheep genetic, like wool quality traits or meat traits?
Yeah, that's right.
So I guess that's a benefit of this project is with AMSI, they are recording this information. So we do have the ability, once this project is complete, looking at the sperm quality traits, we can and potentially could go back and look at how it does link to the quality traits beyond semen quality, which is a great opportunity. I mean, there's so much data that's involved in this project and so much opportunity that we have to explore different relationships to fertility. So yeah, there's definitely an opportunity for that. We obviously need to complete the semen assessment stuff first before we can go on to that. But yes, the scope is big, and that's the great thing about this project is with so much data, there is so much impact and so much you can do with what you have. Definitely.
Yeah. Correct. That's really good. I guess the sort of topical thing that most people are talking about, given the current deterioration of seasonal conditions across Australia, is, do you see any impact of hotter, drier, extreme temperatures on success of AI programs in sheep?
Yeah, so with the environmental data, if you give me 2 weeks, the data set will be completely finished. So yeah, we are looking at temperature, maximum and min temperature, as well as rainfall. And I guess, if the producers see any big changes along that week of AI. So when we combine that all in the data set, hopefully we'll get more of an indication of the range and which like minimum and maximum temperatures we can see and have an impact. And then we can get a bit of an idea and a reference point for the industry to how those environmental changes and if there is any impact at all from temperature or rainfall on AI success. So yeah, the data set is nearly complete and I can nearly give you the answers to that. But yes, hopefully in a couple of months, we can get a better idea of the actual standard reference to the environmental impact to AI.
Yeah, that'd be great, because I'm sure it's a question a lot of people are sort of thinking about, and if it led to sort of improved recommendations for managing the ewes and the rams prior to the event, I think it'd be really worthwhile.
Yeah, definitely, definitely.
All right, so would you just mind stop sharing your screen for me, please?
Thank you.
So we'll there's no more questions, so we'll just come to an end, bring this Let's Talk Rams webinar to an end. So thank you everyone for participating in the webinar. And as I mentioned, I routinely record all the sheep reproduction strategic partnership webinars, and we load them up onto the partnerships website. So there is a resource for people to use and look at again at their leisure. And I'm sure that Simon and Eloise will be happy to answer anyone's questions in the future if they came up. So again, thanks very much. And as I said, we're getting this series of webinars. We're going to build on some momentum. And so our next webinar for the reproduction program will be at the end of October on Friday the 27th. And that one's all about managing triplet bearing ewes for success. So one of the things we're finding is with our industry adoption programs, we're finding that scanning percentages in our really best practice managed commercial flocks are increasing, and that's delivering an increased number of triplet bearing ewes. So the next seminar will really focus on best practice management of triplet bearing ewes and how we can improve survival of the ewes and their lambs. So that will be coming up at the end of October. So thank you everyone for participating today, and I hope to see you again in October for the triplet bearing lambs webinar. And again, a big thank you to our presenters. Have a fabulous day, everyone. Bye.